Oh yeah, right.
Thank you, Monique. Got it. So from now on, it will be entirely recorded. Coming back to the topic, an image is a matrix of numbers, and to illustrate that, you can actually zoom in into a single image, and if you zoom in even further, then you can see each pixels as a little square, and each square has a different intensity. So I really want you to have a hands-on on things and do things simply. So in the chat, there is a link to download ImageJ. We're going to use it quite a lot today. So please download ImageJ, install it onto your machine, whether it's a Mac version or Linux version or a Windows version. The link is at the top of the chat. No, it's not at the top of the chat. The link is going to be set. It's there again. I put the link again, install it, and we will actually open it. And I'm going to show you what it looks like. So the task of today is actually open ImageJ, open blobs, a little file, and transform these blobs into a matrix using this function. So I'm supposed to try to do it on my over computer, and you're supposed to be able to see it. Ah, wait. There. So if you open ImageJ, it should look like that. If you open blobs, it looks like this. You can open blobs in the samples. Samples are over there. What I recommend people to do is to actually have a bigger screen than I do because I cannot see the full list of samples. So I'm limited here, but that's okay. I'm gonna deal with that. So when you do open the blobs, this is just an image of not black, but gray levels. What you can do is image, transform, and you can do image to results. And when you do that, you have a new window showing up. And this is basically the matrix of point. And this image, each pixel of this image here in a row or in a column correspond to a certain value here. Very simple, a very simple way to kind of visualize data. Now, in the presentation, I'm asking, so open blobs, transform the image, what do you see? I guess you see a matrix of point. And now if you want to have the maximum of the third column, just to play with numbers a bit, because we're gonna do that quite a bit. Then you go to your result here, and you're gonna say, file, results, distribution. You have a new window, and you can select the third column or whatever column you want. And okay. And when you do that, you have a new window showing up, and you can actually add the kind of the distribution of this. And you also have the maximum, which is 248. This is an example of how you can actually visualize the number within for an image. So the result using distribution of X3 is 248. And the conclusion of that is that you have an image that is a matrix of points. You can visualize the intensity looking at the numbers. And this is really like a table, like an Excel spreadsheet. So I was doing this presentation yesterday, and it just happened that I landed on this article. I've been working in microscopy for some time, and for me, I always represent pixels as a little square with a number inside. And then I found this article saying that Excel is not a little square 3 times in the titles. And we can actually ask you if there is good people that understand microscopy and they have the answer of why is a pixel is not a square. Maybe at the break, you can come to see me and explain to me why it's not a little square, because for now, I still don't know. No, no. So I read a bit of the answer. I'm not sure about that, but we can actually discuss that. The second point is to actually visualize intensity. So we saw that an image is a matrix of numbers, but I want you guys to actually look at the intensity as a volume or as a three-dimensional volume. So if you, again, open the image J, open blobs, you can actually use the shortcut Command Shift and B to open blobs. And now I want you guys to kind of plot as an interactive 3D surface. So if you go to plugins, 3D, and enter, okay. It's actually not too easy to present in one side and have I should actually split screen. Can I share my two screens at the same time? Can I? No, I can share only one screen at the same time. That's unfortunate. But if the people are located here, all following on the computer, nobody is using the video. So you know what? I'm gonna move to sharing only one screen. And we can actually turn this guy off. So now it's gonna be easier because I have only one screen. Oops. Oops. Okay. I lost my presentation. It's on the other screen and I cannot get it back. I'll be back in a sec. Okay, we'll change methods. Are you guys all here? Ouais. Je vais juste chercher la présentation d'aujourd'hui. So, just to come back. I don't have enough screens. So we were using blobs, visualizing the intensity using 3D projections. And to do that, you go to, whoops. There, you go to plugins 3D, interactive 3D. Sorry for the issue with the screen sharing, the video projector, the chat, and the two computers. So to do that, you go to 3D and you're gonna visualize as an interactive 3D plot. So what is actually this showing is actually showing this image, but instead of seeing intensity as an intensity, you can visualize it as a depth. And you can actually move around and you see that you have white dots at the top. Each dot is a pixel, and you also have black dots on the other side. And you can actually zoom in and out from this. You can increase the grid size and you can scale it. And what I really want you to know is that this image is not only intensity, it can be plotted as a surface of a volume of something. So you can have fun with this 3D plot, but what I want you to have is this intensity of depth within the image. So of course, we can put these numbers as numbers or as intensity, but we can also put them as a depth, something that is volumetric. And this brings me to the bit depth of an image. An image can be encoded into different levels of numbers. So in the volume I showed before, you can have actually two levels, 1, 0 and 1, 1. It is going to be a 1-bit encoding. You can also have 2 bits encoding, which is actually 4 levels. So you have actually 4 levels within your depth of image. And you can have more than that, 3 levels. When we talk about bits, it's actually the number of bits that the computer is encoding. So just to show you, 1 bit is actually 1 value. You can have a 0 or 1. 3 bits, you have 3 values. It can be all 0, it can be 0, 1, 0, and so on. And so you have 8 combinations. So in a 3 bits image, you have 8 values that are possible. And then it increase 4 bit, 5 bit, 8 bit, 16 bits. And the number of values of depth, of levels you can have in your 3-dimensional image is actually the square of this, of 2. So if it's a 2-bit, it's 2 square 2, so it's 4 values. If it's a 16-bit, you have 65,000 levels. In the microscopy, we use a lot 8 bits, 10 bits, 12 bits, and higher. And most of the time now it's 16 bits images, so you have 65 level of grays. But we also use the 1-bit because it's very convenient if you want to say yes or no into one image. We don't acquire image with 1-bit, but we actually use it in the processing. This is actually called a binary image because we have only black and white. The take-home message from this is that an image is a matrix of points, and there is a depth, bit depth that encodes the number of levels you can have into your pixel value. The retina, your eyes, can actually discriminate quite a number of gray level. But actually, there is a controversy of the numbers you can actually discriminate. So I made an ocular test for you. So I'm gonna put this in the chat, this link. I want you to click on the link. Oops, where is the chat? Is here. Anyone knows where is the chat? Here. It's at the top of the screen. There you go. So if you click on the link, you're gonna reach a web page from the Time magazine. Very interesting web page. I'm gonna do it and we can actually compare the result. So you scroll down and you have these little squares. And so you adjust the brightness of your computer to the maximum. You look at the squares and you click all the squares that are not the same color than the background in the middle. So for me, if I look at this image, it might be, don't do it do it on your own screen, on your own web page. But for me, I see that this one are darker. This one, and this one looks brighter to me. And then maybe this one, I don't know. And then when you're done clicking it, you can click submit. You're gonna repeat this 8 times. And at the end, they're gonna tell you how many level of gray can you actually discriminate. So I'm gonna do this with you. Phew. When it gets darker, it's kind of harder. And then when you have your result of the level of grays, you're gonna just put it in the chat. Yeah, I got 37. So my eye can discriminate 37 different levels of grays. Your turn. CV 34, not bad, not bad. 33, 41. Whoa, Marine. 47. Are we going to reach the 50? Actually, it would be cool to that someone gathers all the numbers when everybody has entered the numbers and make the average and the standard deviation of that. So we can have actually real data based on I don't know, 50 people that are there. So in you can continue playing with that. I thought it was cool to kind of do a test to actually answer this question that is a controversy. But the bottom line, what I want to say is that if I don't want the chat, I have to close it. Okay. What I want to say is that your retina is able to discriminate between 30 and 50 shades of grays. And so if you put it in this map, can I have my pointer? If you put it in this graph, then you see that the retina is in the between here and here. But I told you that in microscopy, we at least use 8 bits and even more. So the question comes when you know that some scientific camera can have 700 times more than 50 level of gray. So how do you appreciate something you can actually see? Well, this is where a bit of processing and displaying comes in. Oops. So if you go back to ImageJ, and you open this time another sample called HeLa cells. So you can go to File, Open Sample, HeLa cells. And the first question is, what's the bit depth of the red channel? And to show you what it looks like, I'm gonna do it with you. I can actually close this guy, as we don't need it. And this I can close. So if I go to ImageJ, open samples, HeLa cells, you see that you have 3 colors, the blue, the green, and the red. And at the top of the image tells you it's a 16-bit image. So the red channel is a 16-bit. So now we're gonna do adjust the channel to a 16-bit level. To do that, we're gonna go to Image, Adjust, Brightness and Contrast. And there is a shortcut for that, so I use it a lot. And then you're gonna click Set and 16 bits. And okay. And what happens now? Anyone wants to talk a bit? I feel lonely. Well, I guess it's all black. So what I want you to see is that 16-bit images is way more than what you can appreciate. And you can actually modify the brightness of a contrast of an image so you can actually appreciate it. So the data is still there, it's still inside the image, but you don't see it. And by adjusting the contrast and the brightness, the computer lets you appreciate the image so you can actually use it. To go back, there is a kind of a shortcut to make you appreciate this image. It's clicking auto. If you click auto, it's going to adjust automatically the channels you are looking at into the best settings for a human eye. If you think it's too bright, you can always adjust the maximum and the minimum here. Important things is that this change here doesn't change the value of the data. The data is still there. But you can actually modify it. And this is a bad thing to do because you don't want to modify your raw data. To modify it, you would have to click apply. And when you do that, it actually modifies your data. So be very careful when you apply something to your image because this is not reversible. You cannot come back to something different. So if I click apply, it will actually warn you, oh, be careful, you're going to modify the value of your data. And you see that this graph has changed because you actually changed the value of each pixel. So be very careful when you use the brightness and contrast. It's okay to adjust it, but it's not okay to click apply. A very important concept in microscopy, it's the histogram, or what I call it, the intensity distribution. It's the definition is the number of pixel, or the description is the number of pixel for a gray level, or given gray level. And this curve is very important for you to know. And that's why I'm going to spend a bit of time just to explain it. If you understand this graph is going to help you a lot for the next steps. The first one is what is in the X and the Y axis. In the Y axis, you have the number of pixels. So this is 0 pixel, and this is the maximum number of pixels you have. It's 25,000 in this case. So I have no pixel, I have a lot of pixels. On the X axis, you have the gray level, the number that are in the matrix. So you have black pixels, close to 0, and very white pixel. And here you see that the maximum is 255. And when you do that, actually, when you know that this graph is showing black and white and number of pixels, then it's very understandable how to adjust luminosity and contrast. It's basically taking this guy here, and instead of being here, you're gonna bring it down to where you have actually a pixel. Because here we have very, very little pixel. You see that it's almost 0. So you bring down the brightness, and this is actually increasing the brightness. So this curve is very important because it shows you the number of pixels for each gray level. So if you add the number that are on each line, altogether, the total is the total number of pixels available in your camera. Here, on the top right, you have the maximum bit depth of your camera, or whatever image you are looking at. So here you see that it's 255, and 255 correspond to a 8-bit gray levels. So another function that I think is cool in the ImageD, it's the histogram function, and I think we can use it to look at cells in different way and have this graph displayed in a better way than the brightness and contrast. I'm going to show you how it looks like now. So if I have this image that is still open here, I can see the curve here in the brightness and contrast window. But if I go to analyze and I click histogram, yes, it's going to show me a graph of the histogram. And what it's cool is you can actually click live. And when you click live, it's going to show you the red or the green, depending on which channel you are. And also, if you gate, like if you draw a small region, it's going to give you the curve depending on this region. So if I go in the background here, you see that all my pixels are very dark. But then if I move to a place where it's actually, if I have more cells here, you see that the graphs change and it's much brighter. So this kind of give you a dynamic histogram, and I think it's very cool, and it lets you appreciate your image better. So that was for the histogram and the distribution of intensity, and also the dynamic histogram. The next thing is a lookup table. So I show you how to change brightness and the contrast, but you can actually change a lot of things. If you open Blob again, And if you go and change the lookup table, you can see that the lookup table is just adding a new layer to display the image. It's actually a feature that helps you appreciate the image in a different way. So I'm going to open Blob again. And this time I'm going to go into lookup tables and I'm going to click cyan. So I go from a black and white or a gray level image, but it's still a gray image. The values are still, the same, but just I display it between black and cyan. You don't have to do it between black and a color. You can also have a range of color. So if you look at fire, for example, you see that now it's purple in the dark places and it's very yellow in the bright places or even white. So this is a way just to show and let you appreciate the colors or the intensity better. We can discriminate 50 level of gray, that's okay, but we can discriminate millions of color. So our eyes is meant to actually appreciate colors more than gray level. So I encourage you to kind of use the lookup table to better understand or visualize an image. Very important thing about the lookup table is that the lookup table, why am I always in the middle? The lookup table doesn't change the data. It changed the way it displays the data, but not the data itself. So it's just another way to kind of visualize the image. If we look at the same distribution or histogram graph, and we want to plot the lookup table, the lookup table will just change what color will it be displayed with. So instead of black and white and have gray level, it's going to be black and cyan. But also I can put random colors that I select myself. And I show you the fire, which is a very useful tool for visualizing color, but you can actually choose whatever color you want. And for example, if my cells are positive here, I might decide, okay, this region should be red and all of the rest should be white or blue. And that's what I've done here. So lookup table is changing the display, but not the data. Yeah. And then this brings me to the fact that an image is a multi-dimensional image. So you can see the X, the Y. I also show you the intensity that for me, I consider as a depth of the image. It's not a physical depth. It's not like a Z stack or something like that. It's really a matter of values or visualizing the values. So X, Y, and intensities. But because of the lookup table, you can also add different colors on it. So they are not real colors, they are lookup tables colors. But in this case, I said, okay, I'm going to put a green and a blue and a red and just overlay each of them on the top of each other. And this is actually called channel. So you can have different channels into an image. If you want to navigate the dimension of an image, maybe you can open the file called mitosis in the sample in ImageJ. And you can look at the dimensions that are there. So I'm going to do that. I'm going to close Blob, and I'm going to close this. I'm going to open samples. Oops. And I said mitosis. Yep. All right. So mitosis is a small file, and you can see nothing. There is two channels that are here. And there is a Z stack that is here. You can navigate through the Z stack like that. And you can also have a time. So here it's the time of a video of the cells dividing. You see that the contrast is not great. So you can actually click auto to kind of display a better contrast. And if you do that, you can actually play the video and you can see the cell dividing and the chromosome being separated. So in this picture, there is several levels for each dimensions. So there is 5 dimensions, X, Y, X, Y, Z, or Z, but you can actually scroll here, C, because there is 2 channels, and also T for the time. I always like to add that the depth of the image is kind of a dimension, because I showed you that you can visualize it as a volume. For me, the numbers itself are a dimension, but this is my... Oui. Prio, question. It seems like you cannot see your Zoom screen. Hi. Oh, yeah. Okay. What's your question? Is it above? What is gray level? Okay. The gray level, it's actually a value that has no dimension. It's basically a number. So in the previous class, I showed that cameras receive photons. They transform these photons into electrons, and then they digitize these numbers of electrons, electrical signal, into a value. And this value is a gray level. So is that answering your question? I guess yes. So gray levels are the value of each pixel, and they are integers. So there is no decimal. Usually when you acquire, there is no decimal on that. So how many dimensions? On this image, I have actually 5 dimensions, X, Y, Z, C, and T. How many levels per dimension? Well, if you look at the number of pixels of mitosis, you have 171 pixels in X, 196 in Y. We have 2 channels, 5 stack, like a depth stack, and 51 time points. Plus it's a 16-bit images. So you have all of that combined. Then I have a question, because I know that most of you are beginner or intermediate levels, but there is few of you guys that are actually really good, and they know microscopy quite a lot. So I made a question for changing a dimension. Sometimes it's easier to visualize a dimension in a different dimension. For example, sometimes it's easier to visualize Z into the x-axis, or it's easier to look at time over X axis. So I'm asking people who know a bit about ImageJ and how to kind of change dimension, if they know how to do that. And in a couple of slides, you can raise your hand or talk to me in the chat, and you can give me the answer to that question. To come back to the scientific images, there is many dimensions. The first one is a X. This is what we call a line scan. You can have X and Y, which is a typical image, X, Y, Z, which is a stack, X, Y, Z, and T. This is a time-lapse stack. And you can add colors on the top of that, and this is a channel. Every time you add a layer or a dimension, the size of the file increase. Here, the 16-bit file, it's 2048 by 2048 pixels. And if you do a 3-channel Z-stack time points, then you get this 14 gigabyte file. So it increased quite fast. But you have the option of making all of that a lower resolution if you need to. Important thing is the combination between X, Y, and Z doesn't have to be in this order. It can be, you can do a X and T, or you can do a Y and T if you'd like. So the usual dimension that we use in microscopy on the top of the X, Y, and the one that are presented here are also the XT, which is a line scan, basically over time. XYC, which is multicolor images, and XYCT, which is a multicolor imaging, but over time, so time lapse. Because there is different files format for colors, I want you to play a bit with colors, and for that I want you to open the neuron sample in the ImageJ. This is a 5-channel image, and you can navigate this 5-channel. To navigate the channels, you can use the channel tool, and I forgot to write down where is the channel tool, so I'm gonna tell you where is it. So open sample neuron. So you have your neuron here. You can see that it's all dark for me because it's a 16-bit images and I didn't adjust the brightness. But if I do adjust the brightness for each channel, then I can see my image. The second thing, it's to go and open up the channel tool. It's in color, channel tools, and there is a shortcut for that. So I usually use the shortcut. These channel tools allows you to visualize each channel individually. So if you select grayscale, for example, you can go to each channel and look at them in a grayscale. Why I like to look at them in the grayscale? Because your eye doesn't see colors in the same sensitivity, with the same sensitivity. So if you look at something red and then you look at something blue, you're gonna always say the red is brighter than the blue, but no, you see better the red than the blue. So if you wanna compare intensity, it's always better to look in the grayscale or a fire lookup table. You can look at colors individually because there is a lookup table assigned to each gray level image. So you see that this one is red, this one is green, this one is blue and cyan, and this one is a DIC image. And you can combine them. And this is called a composite image. So when you do a composite, it's gonna overlay both colors on the top of each other. So here I selected the red and the green, and I can see the overlap between the two. Yeah. So this is always the same multi-layer image. I just choose to display different lookup tables to look at the colors, but the image itself is a multi-layer image. Now I'm gonna put it there. I'm gonna put it there. What I want you to try is actually create an RGB image. In the real life, we're working in RGB, and most of the images you found on the web are RGB image. They are not multi-layered channels. To make a RGB image, you select your channels, you go to more, and, or actually, I'm going to select all of them. You go to more and create RGB image. And you see that now I don't have a channel anymore. I cannot slide channels. They are all merged into 3 colors combined together. This is the format we use most of the time in internet because our monitors, or when we want to print something, it has to have the 3 colors that we see the most. So this is the RGB image. When you do that, you go from a multi-layer to RGB, and you want to come back, you can use the split channel option or split color. So if you go to adjust image, colors, and split channel, you can split the same image you had before into 3 channels or 4 channels in this case. But now you see that all my 3 channels, the red, the green, and the blue, they look very, very similar. It's because we merge all these fake colors into one single image. A better example, I'm gonna close this up, is to remove this one. And I'm gonna do the same thing, more, create RGB, and then I'm gonna take this one, and I'm gonna do color and split channels. Because my first one is in red, I'm gonna show it in color. Because my first one is in red, you see that my red is fairly similar to the original red. Because my second one is in green, you can see that my green is fairly similar, but it's a bit brighter. And then I have a blue one, and you see that my blue one is kind of mixed up. It doesn't look the same. I have like this big puncta. Where is this puncta coming from? It comes from the cyan. If you mix cyan, blue, red, and green, then into only 3 channels, then the cyan has to be spread out into all the other channel. And actually cyan is a mix between green and blue. And you see that you have this cyan in the green and in the blue. So I'm gonna close this up and come back to the presentation. The take-home message here is what? The take-home message is that there is different type of RGB images. The one we use in research are multi-layered, and then there is a different bit depth, 8, 12, or 16-bit images. If you combine them into an RGB image, like I did, then you mix colors or lookup tables together. This is okay to do in a figure on a presentation or send by email because you compress the image, but it's not okay for doing this in research. So depending on the usage you're gonna have, you can combine them or you cannot. I just want you guys to be aware that playing with colors is okay, as long as you keep them separated. Once you merge them, then you cannot get back. This is actually there is different format of pictures you can use. Everybody knows about the JPEG. There is a new format that have been released recently. There is also the TIFF that we use a lot, but there is a lot of different image format. Some of them actually modify the data. We call them lossy files, and JPEG is one of them. And this exercise, it's actually to show you that a JPEG image changed the values. So I don't think you have to do it. We can do it quickly together. It's gonna be easier, and it's gonna give us more time for the next part, which are, in my view, more relevant for you guys. So the exercise was to open blobs like that, to transform it, image, transform into results, and to save these results as blobs in my desktop. Now I'm going to take this image, I'm going to save as a JPEG image. So I save it here and I open it up. And then I'm going to say image, transform, image to results. And what you can, oops, second result. So I'm going to keep this open. So if you open this and you compare the first data frame to the second data frame, you see that the values are different. This is 40, here I have 214, this is 223, this is 32. So the JPEG changed the values. If you are wondering why they changed so much, it's because this is an inverted lookup table. So we are looking at something that is bright, but we display as a black. And when you save it in JPEG, it's actually switching this back. So that's why the value are so much different. But actually the value itself also changed. So even if we're inverting it, it will change the value. It was a little exercise to kind of show you that there is some file format that you should be aware that they are compressing the data. Just to give you a brief about the file format. So there is the lossy ones, and JPEG is the mostly used. It's not a bad file format. It's just that you lose data when you're using it. The good news is that you compress it. It's easier to send. But the bad news is that you cannot do analysis on it. Then there is a lossless formats, and usually the proprietary, the company that are making microscopes and things like that, they are lossless formats. And there is also the TIFF, which is a lossless format. Example of different kind of file format you can find with the company are LIM, ND2 for Nikon, Carl Zeiss Imaging, OIF for Olympus, LIF for Leica. There is also like open source formats such as TIFF, JPEG, and GIF or GIF. I don't know how you pronounce it. But the one we recommend, it's actually the one that are normalized for microscopy. And this is called OME TIFF and is made by Omero, or the Open Microscopic Project. It's basically a TIFF that has data that is structured inside. It's not only the pixel, but you have also something we call metadata. Just to give you an example of what metadata looks like, you can open up again in ImageJ the neuron, and then you're gonna click Show Info. So if you go to Image, Show Info, Show Info. So open sample, neuron, and this is what you see. You have a new window, which is actually a text file. This text file is saved within the TIFF. So you can bring it up whenever you need. If you read the text at the top, you actually learn a lot about what is this image coming from. And it tells you, for example, that this is a confocal image of a hippocampal neuron. And it tells you what is in channel 1, channel 2, channel 3, channel 4, and so on. There is other data that is shown in this file, and these are important data to actually understand your image. And this is actually the number of pixels, 512 by 512, in the image. And you see also that you have actually dimension in micrometers. And these dimensions are actually a real dimension of the image. This dimension is calculated based on the resolution, pixel size, which is a 6.25 pixels per micron. So if I know that each 1 micron takes 625 pixels, then I know what is the size of a micron, of a pixel, and then I can calculate what's the width and the height. A voxel is actually a 3D pixel. It's actually a little volume. The voxel depends on the size of the pixel, but also the depth that the image has been acquired. So this is typical when you are doing confocal microscopy. Then there is the bit depth that we saw, and there is also the display ranges, which is the brightness and contrast that are saved directly into the file. So when you open it up, it will show you directly this value unless you say otherwise. Last thing I want you to see is that there is also the number of channels and the display mode for the colors. So in this case, we are in a composite mode, which is overlaying all the lookup tables on the top of each other. There is different kind of metadata, and the first one, I showed it to you, they are within the image. There is the X, the number of pixels, the size of the pixel, and the bit depth. Others are related to the series of images. So for example, if I took a Z-stack or different channels, I have this saved. There is also metadata corresponding to the instrument, such as the power or the exposure time. And finally, there is metadata corresponding to your sample. I have a good news for you is that nowadays, most of these metadata are, most of these metadata are within the file saved by the company who are making microscope. So all of this is already in the file. You don't have to take care of them. The only thing you have to take care is your experiment. Is it a control? Is it a test? Is it a drug? And what I do, and this is my tip of the day, is to include this metadata within the name of the file. If you do that, then you have all the information that is required to analyze your file within the file itself. And an example of that, it's you put the date, you put an underscore, and then you put, okay, that's my condition 1 where I treated with a drug, I don't know, like 1 dose, and this is my sample number 1, and then I repeat this 3 times, and I change the quantity of the drug and so on. If you do that, then you have all the files that contains the experimental metadata, and the file itself that contains the instrumental metadata. You can also add metadata on the go. And this is a very easy example. And I show it to you and give you an example because it happens most of the time that people are asking me, how do I put the scale bar on my image? The first thing is you don't put scale bar on your image until you're publishing an article. So you just need to put the resolution or the distance within the file. And to do that, I'm going to show you an example. I'm going to close all of this. I'm going to open the sample called leaf. And you see that this sample is not calibrated. We don't know what's the pixel size. And I know that directly because here it's written in pixel. If ImageJ has the data, it will tell you actually the real values, the real size. So I'm going to zoom in into my image. And I'm going to take this line tool, and I'm going to measure maybe from here to here. I'm going to draw a line like that. And I know that in real life, this corresponds to this distance. So it's 85 millimeters. So it's 10.3 minus 4.6. So it's 85. Now I want to know how many pixels do I have here, and actually this I can read it directly. It's about 507, or I can measure it, but I can read it. It's the full length. I could have done this differently. I could have measured this, which is 1 centimeter, and I can measure the number of pixels by just clicking measure or analyze, measure. Oops. If I do that, I have a result window showing up, and this tells me the length, or should tell me the length of the length. You see it's 60 pixels. So if you divide now 1 centimeter per 60, then you get the scale of the image. The good news is that you can actually add this data directly into ImageJ, and it's going to be saved within the image. So to add the scale, or the scale, not the scale bar, but the scale of the image, you just go analyze, set scale, and then you're going to put the pixels that you measured. I think I measured 60. And I know that the distance was 10 millimeters. The pixel aspect ratio is always 1 now for most modern cameras, but it can be different for older ones. The unit of length is millimeters. So I have 6 pixels per millimeters. And I click OK. And now you see that the image is calibrated. I have my values in millimeters and also my value in pixel. I don't need to add the scale bar right now. If you add the scale bar into the image, you will actually lose the data that is behind the image. So just keep the image with the scale integrated. When you're gonna make the figure, you're gonna add within the software you're using to make the figure on the top of that, the scale bar. So you don't lose any data, and you can actually change the scale bar if it's too long or too small or at the wrong place. You can actually modify and move it. So to come back, you can actually change the metadata yourself, and one of them that is very important, it's the scale of the image. It's giving you it's giving you a link between the size of the pixels and the size in the real life. If you don't put the scale, you're gonna have measurements made in pixels. If you have the scale, then the ImageJ will give you automatically the measure into the value you are entering. If it's millimeter, it's gonna be a millimeter. It's microns, it's gonna be microns. If it's Angstrom, it's gonna be Angstrom. So you don't need to add the scale bar into your image right now. As long as the image is calibrated, then you don't need to add the image. The good news is if you go to a microscope and you see that all your images are not calibrated, and you take the image the same way, then you can just add this scale bar in batch, and this we can do it at the end of the session in the third part. Very big question is, which format should I use for my images? This really depends of the usage. I don't say anything good or bad about each format, but in research, you use the proprietary format for the acquisition. Why? Is because the companies are making links between the instrument and the metadata. If you want to keep most of the metadata, then you use their format. You know that it works best. After the acquisition, you can actually switch to something that is not proprietary, and TIFF or OmniTIFF are the best way to do the processing and the analysis. If you want to use RGB, you can do, or actually you should do, but only when you reach a point where compression is okay. Like I don't need to do quantitative analysis on my image anymore. I'm gonna keep the original image, but I'm gonna create a compressed version, an RGB version, to send to my boss or to make in my presentation, so it doesn't go too big file. So the conclusion is, all what I've said, I can repeat them, but I put this slide up because I want you guys to have one slide to memorize or to come back to if you need to just to refresh your memory. You don't have to read the full presentation. You can just go into this thing. And now it's 10:04, so I'm exactly 59 minutes after start. So we're going to take a small break, but before that, I'm going to take questions. And I want to thank Monique Vasseur because she helped me a lot with this, making this presentation, giving me feedback. And she's actually the source of most of this workshop, because at the biochemistry department, they give un cours siglé, which you can actually register to get, and they give most of the information I've given, but they take a bit more time and they are more professional than me. So if you want to register for this class, you can contact Monique in the Zoom, and she will let you know what is the class. Thank you. And I'm going to stop sharing for now. Any question in the chat or in the real life? No. Remember my second slide, which is, it's your workshop. If you want to make a picture, so question from Francois, if I want to make a pretty picture, can I use Photoshop? No, you can actually use Photoshop to make a pretty picture. There is some ethical aspects of image processing, and there is few rules we need to follow in a scientific imaging. But if it's for making a cover of a picture or of a journal, if you would just want to have something pretty, you can actually modify your image. We are in a different field. It's not research anymore. It's art. And I think modification of image is totally fine. We just don't want to draw conclusions from your modified picture. You just, it's artistic view off. It's not data off. So there is we'll see in the second session, the processing. We do a lot of modification on images. A lot of them are fine to do, but some ethical aspects tells you cannot modify a sub part of an image. If you modify an image, you modify all of it. You don't modify a sub part. You can crop, but you cannot crop to hide something that goes against the conclusion you are drawing. If you process some images one way for your control, you have to process the same way for your mutant. You cannot change the image in a way it will change the conclusion. A very big ethical aspect. So Photoshop is fine, but just for illustration. Also, the filters we are using in ImageJ and other softwares, you can find them in Photoshop. They are exactly the same. Any other question? No, then you have 3 minutes, 5 minutes, five minute break. Bonjour, on est de retour. Je vais essayer une autre technique pour essayer de partager mes écrans sans que ça soit mauvais. Donc, le premier écran est partagé. Le deuxième écran, quand j'aurai besoin de le partager pour montrer des choses sur ImageJ, ça va faire ça. Et là, on passe à l'autre écran. Est-ce que ça fonctionne? Là, vous allez voir quand je vais faire les tests avec ImageJ, les choses comme ça. Donc, voilà. Hop. Ah, il faut que lui, ils arrêtent de partager alors. Et voilà. Est-ce que vous êtes tous là? Tout à l'heure, je parlais de changer les dimensions, de visualiser une image, de changer les dimensions. Est-ce que quelqu'un veut regarder ou présenter ça? Est-ce que, par exemple, Alex dans la liste, ou Nicolas, ou des gens qui sont un peu avancés en analyse d'image, ou Elvis aussi qui sait faire ça, j'imagine. Est-ce que tu sais changer les dimensions d'une image? Est-ce que tu veux nous présenter ça? Passer par exemple du Z en T. Est-ce que Monique, tu veux le présenter? Tu sais faire? J'imagine que oui. Hein. Et s'il y a quelqu'un d'autre qui est volontaire, qui veut faire un peu du peer sharing, montrer comment changer d'une dimension, passer d'une dimension à l'autre. Alors, je vais le faire par exemple. Donc là, vous avez mon écran. Vous avez, on va prendre le mitosis. Voilà, on va prendre le mitosis. Donc, des fois, c'est difficile d'apprécier des choses qui sont dans une dimension parce qu'on a du mal à les apprécier dans le temps. J'ai montré comment. Oh, I switched to French. I showed to how to switch dimensions, because this is something important. One of the dimension we can switch is actually intensity. You can change from gray level to color, which is actually a new dimension for us. You can also change, and I show how to visualize color in 3 dimensions as a volume, and this is also a change in dimension. But then you can also go to your stack here and do an orthogonal view. And when you do that, is basically you are plotting the Y against the Z or the X against the Z. And when you move the cross, you can actually appreciate this different view. You can also move it through time, and then you can maybe see better that here the green is not overlapping with the red, and here the green is not, which is something you don't see very well here. You can also do in your stack, image, stack, and you can do plot a Z-axis profile. You can choose the profile you want to see, if it's the time or the Z. In this case, maybe we can plot the time. And this is actually the intensity over time that is decreasing. You see that your tissue is bleaching progressively. So this is another way of changing dimension to visualize something in a different dimension, because it's very hard to appreciate if the intensity decrease here, but in a graph like that, it's going to be very easy. Also, this can be done in live mode. And I guess if you make a specific area, or maybe it doesn't work in orthogonal view. So these are examples of changing dimensions so you can appreciate things better. Yeah. So go back to the second presentation. So the first one was about the digital images. The next one is going to be about processing images. So image processing is a very important step to kind of be able to analyze your image. And to do it well, you have to do it in several steps. It's actually having 3 different steps to do the image processing. The acquisition, we discussed it in the first session. So this session is going to be image processing. And the 3 steps we're going to look at. The first one is the pre-processing. The second one is the segmentation. And the last one is the extraction. The pre-processing is basically everything you do to facilitate the second one. The second one is actually making several sub-regions within your image, depending on what you see on the image. And the extraction is actually getting numbers from the region you drew before. We're going to also see the statistics and the analysis, but this is going to be in the next session. So for the pre-processing, there is different methods or different kind or type of pre-processing you can do. The first one is correcting intensities. So this actually modify the values of your data. The second one is modifying the position in X and Y or Z. The third kind of pre-processing you can do, it's filtering. And then there is also the Fourier transformation, which is made by this guy, Jean-Baptiste Fourier. It's a very interesting way to kind of look at images. And I just want to give you a sneak peek of what it is. Maybe some people will say, oh, I've never seen this before. The first thing I want you to do is actually, the first thing we're going to do, it's about correcting illumination. This is a very usual things we see is when the light coming into your microscope or to your image is not ideal or it's not perfect. And then you need to correct for this. And we're going to actually do this together by opening image J and opening the sample called cell colony. And to have a look at the issue, we're going to display the color lookup table called 16 color. And I'm asking you guys, what is the issue? So I'm going to do this here. So I'm going to share my screen and I'm going to open up which one colony? Yeah, cell colony. So you have this image here. And it's hard to appreciate if there is a darker corner. I can see a bit it's a darker here, but if you look at it with a lookup table, it's going to be easier. So I'm going to use the 60 color. And now that the intensity or the brightness is encoded into colors, I can clearly see this is red and this is white. So there is an issue and there is a gradient here. So that's the issue. The issue is that the illumination of this colony is not ideal, and we need to correct for that. So how to correct for that? Well, the first thing we're going to do is duplicate the image, because we never work on a single image. We're going to duplicate it. And then we're going to create a background image. And to do that, we're going to look at this function, process subtract background. So you can, in the pop-up window, you can check the light background, because it's a bright field image. So the background is bright, it's not dark. So you can click light background. You can also click create background. Instead of subtracting the background directly to your image, you're going to create a new background, and then you're going to click preview. And then I'm going to ask you to play with the options to look at how the options modify the results. And if I do that, I'm going to duplicate the image. And then I'm going to go to process, subtract background. I'm going to check light background, create background, and I'm going to click preview. You see that it made this kind of a density map showing the brightness. This is very similar to what you see at the back, but it's usually better to click sliding parabol. This is more accurate. You can also disable the smoothing if you want something more that is more accurate or less broad. And you can actually change the value of the pixel. If you put something very small, you see that you have a lot of details. Maybe I don't want to go into this kind of detail because I know that my illumination is not that different. It's maybe something like 22 seems something reasonable. So if I click OK, then I have my background and my cell colony side by side. And what I want to do is subtract one to the other. But if I do subtract one to the other, and you can do this with the image calculator. So you go to process, image calculator. You're going to select the first one as your image and the second one as the colony, like the background image, and you're going to select subtract. And I'm going to click create a new window because I want to keep both of them. If you click OK, and then you have a black image. Something went wrong. I'm going to look at the distribution because I don't see anything. So I'm going to look at the histogram, and you can see that the histogram is 0, 0. There is 0 everywhere, actually. The mean is 0, the maximum, the minimum is 0. So what happened here? Well, it happens that if you do a subtraction between these values, and you can see the value in ImageD that is displayed here when you are pointing on it, here it's 245. It's a very bright pixel. If I remove 245 at this pixel, which is 232, then I'm going to get negative value. And because images cannot be negative, then I have everything black. It's all zeros. So I'm going to do it again. But this time I'm going to process image calculator. I'm going to do the same thing, but this time I'm going to click 32 bits float results. And if you do that, click, then you have a very nice, evenly distributed image, and you can still see your colony. But now when you point into something, you see that the values are -11, -31. When you select 32 bits, you can actually have a negative and also decimals values for pixels. So instead of having an image with finite numbers, you have an image which is kind of infinite numbers of depth. So this is made for being able to do calculation and also having negative values. Fortunately enough, there is a way to do this kind of thing without having negative values. And this is by doing directly the process subtract background option. So instead of creating a new background, I'm going to say don't subtract. And I'm going to click preview. And now you see that I keep the same options, but now you see my image is corrected. And I click OK. And now you see that the values are 245, 246. It doesn't go into minus something. Because when you do subtract background directly, it actually, the calculation is a tiny bit different. And I'm going to show you in the presentation what is different. So we did the illumination correction, very important step. And I'm going to show you, these are the options. This we did. Also, I explained what is 32 bits bit depth float. And also I showed you that if you do it directly, then you don't have these negative results. If you do the background subtraction, you know, the intensity corrected equal the original minus the background, you might have negative results. That's why you use 32 bits. But if you do on a pixel to pixel basis and you multiply it by the mean of the background, then you don't have. So there is kind of a ponderation here, a mathematical adjustment that we do to kind of avoid having negative values. Another example where la correction d'illumination or illumination correction is useful is also when you have dust on your light path. So here you have the image that was it's about DIC, and you see that you have cells, but there is dust a bit everywhere on the camera. You can actually calculate what is the background of this, and you can subtract it, and it's very good to remove all of these artifacts. Is this okay to do that? Yes, as long as you have this picture. There is two ways of having this picture. The first way is to generate it, and we did. The second way is to actually take a picture of an area where you don't have samples. So you can do this. This is the best and the most accurate way to remove the background, is to take a picture of something where you have no sample, and to take the same picture with the sample and to subtract both. So the light intensity corrections are very good to make images look the same. So if you have images that are illuminated differently, then they will look the same at the end. You can also normalize intensities with this. If you have a brighter and a darker, you can adjust them so they look the same. And you can also correct the illumination if it's not, if your field of view is not perfectly illuminated. You can also suppress artifacts, and there is different kind. And I showed about the correction of illumination here. This is an example of a tiling. Is when you take 4 pictures and you tile them together, you stitch them together, and you see that if you don't have a correct illumination, you can have this kind of band that appears in the between. If you want to avoid that, you have to do the shading correction or the illumination correction. The second pre-processing we can do is a geometric pre-processing. It's if you want to orient or align images in the same direction, this is very useful. Also, if you want to stabilize a video, if you know that something is making your image moving and you want to stabilize it over time, this is usually geometric transformation you can do. You can increase or reduce an image. Also, this is called scaling. You can rotate, translate. One important thing is about registration and tiling, and this is an example of this registration of tiling. Imagine you have 2 images taken at different magnification and also at different orientation. With this one is color, this one is black, and I want to overlap them. You can see you have to rotate, you have to translate, you also have to kind of magnify a bit this one, which is a bit smaller. And at the end, you can have 2 overlapping images. This thing is it's made using a registration. For example, on this image, you're gonna say, okay, this is my corner here. This is a chimney. So I'm gonna draw a point here, another point here, another point here. And I'm gonna do the same thing on the second image. And then you're gonna stretch or move the image to overlap the second image. This is a process called registration. The tiling is the one that I showed you before, is when you take several images and you stitch them together like a little tiles. Here you can see that you have clearly 6 tiles by 6 tiles, and you have this shading that appears. And if you don't want this shading to appear, you have to correct for illumination bias. Yeah, so registration, tiling, and correction of illumination is important. There is different kind of transformation. I didn't talk about. This one are kind of fixed because you don't stretch your data, you don't stretch your image, but you can have also things where you actually stretch part of your data. And this is useful for big tissue or brain images or things like that. In microscopy, we don't use it often, but it's something I wanted to show it to you. Another pre-processing, I'm going faster because I want to spend more time on the second part of the session where we actually play with our own data. And this is just to show you that you can put filters on images, and this actually help you to do the segmentation. These filters help you to remove noise, find the edge of an object, and there's many more. And there is different also kind of filters. You can have low pass, high pass, Gaussian. This one is a Gaussian blur. You can have convolution filters, erosion, dilatation, and other things. And I know that some of you guys are already writing micros, or they know a bit of ImageJ. And I was wondering if one of you could actually write a little macro to test on blobs the different filters that are available in ImageJ. I actually didn't find it on the web, anything that was giving you a clear access, a possibility of what are the filters. It would be useful to have a small script or macro that can actually show you what are the filters and what the result on a given image is, on a given image. So if you want to keep listening to the talk and at the same time write this little macro, you are welcome, and you can present it in the automation part later on. So I talked about the pre-processing, which is a very important step, and this will be related to automation later on. But this pre-processing does one thing: it's helping with the segmentation. So the second part is about segmentation. What it is? Well, it allows you to actually create sub-regions within your image. And these sub-regions are called ROI, of region of interest. Sometimes they are also called objects. So you're gonna define different objects within your image. There is two way of doing segmentation. The first one is based on the intensity, and this is called thresholding. The second one is, instead of using just the intensity of each pixel, we're gonna use a lot of many variables, and this is called pixel classification. And I'm gonna spend more time. This is a kind of a historical, very accessible tool. This is kind of a new trend, and it's very cool because you don't need to learn a lot about what are these variables. The first one is the intensity-based one, and there is a small exercise here. Is basically go on the web, find some DAPI stain cells, and open them in ImageJ. So I'm gonna do that in my second screen. And then we're gonna adjust the threshold, and we're gonna move the slider to kind of threshold the image and click apply. So if I share my screen here, it just happened that I looked for these cells yesterday, and they are called DAPI cells. And yeah, they look like that. Oops. I really went to an article, the first one that was online. So, so I have my DAPI cells here. You see that you have 4 conditions. So I'm gonna take one here. It's a 8-bit image. It's a bit blurry, but yeah, that's the first one I found. And I guess images are not perfect. So to adjust the threshold, you go to adjust image, adjust threshold. And when you do that, you have to say if it's a dark background or a bright background, and it's gonna show you these red images. Everything that is red is what is kept by the image. Everything that is transparent is what is removed by the image. So in this case, you can move the slider to kind of select what you want based on the intensity. So this is the highest intensity. If I go down, you see that you start having pixels that are selected. This is the brightest pixels. If you still go down, then you have most of the cells. And then you keep going down, and then you get the dark. And the question is, where do I put this threshold? This is a very difficult question, but if you plot the histogram, if you look at the graph at the back, you can see that you have one peak here and another peak here. So usually you want to put the slider between these two peaks. There is also a defined algorithm within ImageJ that can decide for you where to put the slider based on the pixel intensity. And I highly recommend you to use them whenever it's possible, because this is unbiased. If you say, oh, I'm going to adjust it because I can see my cells and I know that I want all of that. And then you go to your controls, to your mutant cells, and I'm going to say, okay, this is my my test cells, or I'm going to adjust it, but in this case, I just want this. So you see, this is a biased, experimental bias, because you know what to expect in each condition. If you use an algorithm, it's going to choose for you whatever the algorithm gives as an output, and you can actually have this for both your control and your mutant. And then you can say, I use this algorithm to do my thresholding, and these are the results I obtained for my my control, and these are the results I obtained for my mutant. When you choose one value, then you can click apply. And when you do that, it's actually giving you a mask, a binary image. So the output of the thresholding is basically telling you everything above this is going to be kept. Everything above below that is going to be removed. And that's what you obtain here. You have pixels that are kept or pixels that are removed. All right, so I'm gonna go back to this. So now you have a binary image, and you're gonna tell me, Nico, so what? I had a very nice image before, and now I have a very not good looking image. Well, when you have a binary image made of 0 and 1, then you can do more math on this. And one of the things that most of the people are interested in is actually counting particles. In ImageJ, you can use that, and in your binary image, you can go and do analyze particle. And what this gonna do is gonna show you a pop-up windows where you can actually select different features. And based on this picture, the software is gonna count things for you. So in my image, the question is, how many cells do I count? And the question you can ask yourself is, how many cells did you count? So I'm gonna share my screen in this. There. So I have my binary image. Sometimes you can clean it up a bit. And I'm gonna show you one way to clean it up. It's to use a filter or binary filter. One is called open, and the other one is closed. I don't remember which one is which, but if you do close, you see that your cell is gonna blow a tiny bit. If you do open, you're gonna see that they're gonna shrink a tiny bit. And these remove very, very small pixels that are not very interesting. Another things that people usually do, it's a watershed function. Is basically this watershed is gonna try to kind of break cells that are close to each other, and it's gonna draw a line like that. So it calculates where are things that looks like a drop, and he's gonna break them in the between. It's not perfect, and some people might find it not very useful depending on the sample, but in this sample it works well. Can I remove the background and then adjust the sample? Question from Usam. Yes, you can remove the background and then adjust the sample. The background removal will be in the pre-processing part. The thresholding will be done into the segmentation part. So the watershed is there, but then you're gonna go to analyze particle. But before you do that, what I want to know is what is the size of a cell roughly. because we have a binary mask, it's very useful to use this kind of magic wand where you're gonna just click on the cells and it's gonna select it, and I'm gonna click measure. You can, to do the measurement, you just go to analyze measure. And this is gonna do the measurement within this little cell that I selected. And what I'm interested in is the area, 558. This is in pixels. You see that the image is not calibrated. And that's all I wanted to know. What is the area? And maybe what's the diameter also. Sometimes it's useful to have the diameter. In your case, you might have less data. In my image day, I display all the data that I can when I do my measurement. Yeah, the diameter is 27. We're gonna discuss about these values later on. So when you do your analyze particle, it shows you this window, and one of them, you see it's various different options. One of them is the size in the pixel square. And I told you that my sales was about 500 something. So I guess if I put something like, I don't want the very small ones, and I don't want the very big one, but I'm gonna give you a range between 20 and 1,000. Then the circularity, this is actually something that doesn't look like a circle at all. And one is when it's perfectly a circle. So I know that my cells are roughly a circle, so I might use 0.5 to 1. This is what you want to see at the end. I might want to see the ellipse, for example. You can also do the analysis and counts, so you can display the results, summarize. What I like to do is also to add to manager. This is cool. I like to exclude on edges because these are not on the edges. You don't have the full cell, so I remove them usually. And when you're ready, you just click OK, and you see that I don't have anything. Oh yeah, why I don't have anything is because I did the analysis on the single cell I selected. So I need to do the same analysis, but instead of having you see a square selection somewhere, I need to do it on the full. So the good thing is ImageJ remembers these values. So if you do OK, then you say, OK, I counted 34 cells and here they are. And the good thing is that they also put them into something called ROI manager. And this is giving you the position of each cell, which is good because after you can go back to your original image, which is here. And oops, no, this is not my original image. My original image is here. And you can actually go back and say, okay, this is one cell, this is one cell. And you can actually check if you were correct or not. You can also measure now the intensity within these cells. And for that, you just click measure. So this is what the segmentation gives you. It gives you several regions of interest within your image. And the idea will, the main challenge in segmentation is to actually do it in a proper way. And this was very easy because the blobs is a very easy image. This DAPI image, I took them because they were easy. But let's do the same thing on this image and you'll see that things get more complicated. So I'm gonna do the same thing. I'm gonna adjust the threshold. You see that cells are touching each other, so kind of hard to kind of find what is the good level. So I'm gonna use the Otsu again, and I'm gonna click apply. It's all a mess here. So I'm gonna do analyze and maybe process binary, and I'm gonna open to remove some of the background. I'm gonna maybe watershed them. You see broke them, but is that actually a good separation or not? I don't know. And I'm gonna do the same analyze particles. And you see that now, well, I have less cells and yeah, the size seems to be bigger, but I'm not very sure about the quality of the data though. So yeah, this can be a challenge, and that's what I wanted you to know is that for some images, these kind of things works very well. For some of our images, doesn't work well at all. But fortunately enough, there is good news. So I counted 33 cells in my image, and what are the limitations? The limitations are it doesn't work for all images. Now I'm going to move on to pixel classification, which is, in my view, what you're going to mostly use in the near future. and hopefully from now on. And this is based on actually a value of a pixel, such as this 48 here. This 48 is not a single number coming out there like that. It's actually 48 here because this is bright, it's an 83, and this is dark, it's a 19. And you have something like you have a fluorophore emitting light here, and you see you are in the border between these values. So the 48 is not alone, and it's not an independent number. It's depending of the 83, it's depending of the 19, and it's linked to that. So it's decreasing to get to 19. So there is a high correlation between pixels among each other. So you can actually do use these interactions and other variables that are linked to this value to classify pixels. And now we're going to move to something called segmentation and trainable segmentation using a tool present in Fiji. It's a very simple tool, but it's actually working very well. So I'm going to move up to Fiji in my other computer, and we're going to open something called trainable Weka segmentation. And you can find it in plugin segmentation, trainable Weka segmentation. So I'm going to share my screen. I'm going to open up Fiji. Oh, by the way, Fiji, F is Fiji, I is just image J. So Fiji is just image J is actually the name of that. I guess you guys knew that. So I go to plugin here and I go to segmentation, and I'm going to open up Weka segmentation, trainable Weka segmentation. When I do that, it asks me to open an image. So I'm going to open an image, and I basically have exactly the same image that I had before. And how a pixel classification work is that you have to train kind of a model, or you have to tell them, okay, this is my pixel that are positive, this is my pixel that are negative, and I want you to find among the image all the pixel positive and negative. And for this, you have two classes. You need at least two classes to train a model. So I'm going to draw, you see a line here, and I'm going to say, this is my background. And then I'm going to draw in a cell, and this is my cell. And then I'm going to ask train. So it's going to calculate a bit. And now you see that he say, okay, all the pixels that are similar to that, they're going to be green, part of my nuclei. All the pixels that are not, they will be the background. But you see, it's not perfect. You see that here, for example, it didn't find the region. So you know what? I'm going to retrain it. You can toggle the overlay by using this. But I see that here, for example, it didn't get that this was background. So I'm going to click this. Oh, maybe another here. And I'm going to say, okay, this is actually background too, and retrain it. Okay, now we understand that this is actually a background, but it also removes some places where it's a nuclei, but you think it's a background. So I have to tell him, okay, actually this is also my nuclei. So you can train it again. And now you have a kind of a segmentation that is not bad. You have maybe one cell that is still outside, but you can retrain it. You can also kind of select a bit more of this, add, and train again. And there you have a good segmentation that is really, really close to reality. So again, if I toggle one and to the other, I have my mask, and you can see that all my cells are kind of detected properly. So this is cool, but you can actually go further. You can actually create a new class and say, oh, actually, I want to segment in more than two. I'm going to say there is actually foci in my nuclei. So if you toggle here, you see that here you have a cell that is maybe dividing or something. I don't know what's going on here. But what you're going to do is just, you're going to draw some pixel here. And you say, okay, actually, this is my third category. And you can train again. And now he's going to say, okay, this is a new region. I identify it. You see that it's purple. He thinks also that this is actually another region that belongs to this. But looking at the image, I see that it's not. So I'm going to say, no, this is actually a nuclei, and train again. And now you have an almost perfect image. you have a model that actually is able to segment your image. The good thing is that you can save it. I train my model into an image, but I can apply it to another image. And I'm going to do that now. So I'm going to apply, and I'm going to apply to this second image, which was from the same article. You're going to create yes. So I'm going to explain to you what's a probability map versus what is a segmentation map. Okay, so this is a probability map. It has 3 channels. The first one, everything in white is what is positively within the category. So my category is background. So you see that you have actually all my background that is white. It's good. The second is actually the nuclei. He actually identify all the nuclei and he put them in white. And the third one is about this kind of clustered foci. But you see that in this image, he identified the cluster quite very well. So yeah, so the segmentation is done and it's done in a proper way. The difference between the probability map is that you have gray levels, white being highly probable, and gray and black being not highly probable. By looking at that, you can actually refine your model. You see that in this cell here, it's white, but it's a bit grayish, meaning that it's not totally sure this cell is actually a foci or nuclei. You can go back to your original image and look at that. And I think this cell is here. Yeah, and you see that the DNA start to condense in these cells and it's a bit brighter. So that's why it's wondering whether or not it's a foci or not. In my view, it would be not. So I might correct the model and train again to kind of correct that. The good thing is you can actually batch this now to apply this classifier on different images. So this one, I'm going to just create the result. So I don't want to create the probability map anymore. I say, okay, that's a good result. So I have my mask. The red is the background. The green are the cells. And the purple is actually the condenser or the foci. I can use this to go back into my image and say, okay, I want to know the intensity, or I want to know the quantity of pixels that are within each category. But this is done in J extraction. But you can see that in using this kind of a very simple trainable Weka, you can actually segment your image quite easily. And it's easier than using luminosity. Okay. Anyone tried that on his own computer? Did it work? Did you try, anyone tried with your own cells? Did it work? Anyone, please answer me. I feel lonely. Ah, il y a une question. François. Okay, how is this selection different than doing by hand? Well, because you don't do all of them, you just do some of them, and then you can propagate them. Also, doing by hand, actually the human eye is very, very, very good at discriminating shapes and finding things. So it's very hard for a computer to reproduce that. But the idea is to be able to create a model and then apply it in an unbiased way or propagate it to the rest of the data.
Ouais, sauf que le modèle, il est créé de façon biaisée. C'est encore François, c'est juste que je parle maintenant.
Oui, je t'entends.
Donc, mais par contre, le modèle de base, il est créé de façon biaisée par nous?
Biaisé, oui. Tant et si bien que quand on regarde le résultat, ça signifie quelque chose.
Mais encore une fois, le résultat, si tu l'as créé de façon biaisée, il va donc être biaisé. Même si la machine, tu lui dis d'apprendre le biais, tu lui dis d'apprendre un biais.
Le biais, c'est pas de créer une segmentation, c'est de ne pas l'appliquer de la même façon à tous les autres. Par exemple, le thresholding, si on le fait basé sur sa volonté personnelle, on va être biaisé à le faire différemment pour le cas contrôle versus le cas test. Le biais, c'est ça. Le modèle, le fait de l'appliquer à tous, peu importe quel est le modèle, il peut être bon, le modèle, on peut avoir une bonne segmentation, ou elle peut être mauvaise, c'est pas très grave, dans le sens où on va l'appliquer à tout le monde.
Okay.
Le biais, c'est vraiment dans l'application plutôt que dans la création du modèle.
OK, merci.
Et oui, c'est du machine learning, ça utilise des algorithmes de machine learning pour faire ce genre de choses. Je voulais juste montrer dans les settings, peut-être, je vais juste repartager mon écran alors, ici. I just want to show you that in the settings of the meta segment, here, you can actually select what are the features that the pixel you selected are including in the segmentation. So here you see that you have only 5 of them that are selected, but you can actually select much more. And actually the mean, you see the mean intensity is not part of the selection. So when you take the picture, it doesn't use the mean as a training model. So you could actually select more of them and retrain the classifier and see if by putting more features, it gives better results. So in this case, I guess, yes, it gives a not bad result at all. Yeah, so just to give you these things, you can combine them. As soon as you start putting them a lot, then it's gonna be a kind of, it's gonna take longer to train the classifier. All right, so the next part is train the classifier and create the binary mask and done. So for me, it's more reliable way to kind of segment your image is to use pixel assignment and pixel classification. There is 2 other tools that are also very interesting doing this kind of thing. In my view, they work as well, if not better, than the Weka segmentation. I'm just going to show you an example of Elastic and maybe QPath if we have time, but it's already 11 o'clock. So I'm going to share my other screen. So Elastic is a software you can download, and it's been done recently. So it's not Fiji and ImageJ. It's another software. It looks like that. The icon is not very, very, very nice, but anyway. So you can open it up, and it actually does many things. It does pixel classification, but also it does object classification. So for example, in my case, I had 3 objects. I have the background, I have the cells, and I have the foci, but it can also classify them if you have many of them, for example. In this case, I'm just gonna show a quick demo for the pixel classification. I'm just gonna take these cells and I'm gonna put them on my desktop. And I'm gonna open my control. Oops. Oh yeah, I forgot to say that Elastic needs an empty folder to work. So you can add your images the same way that we did in Weka. The difference is that you can add more than 1 images at the same time. So this is my input data. My data as metadata. So, oops. So it is able to read them usually. And you can also change, here it's, you can change the dimension just by typing them, which I found it's actually cool. And then you can select the features you want to analyze. So there is features about intensity, edge, and texture. And if you don't want to, if you have a good computer, then you can select all of them. These are the features that the model is going to use to be trained. The more you select, the better or more refined it should be. Then this is going to be my background. This is going to be my cells. And you're just going to paint them. So my background is here. And my cell is here. And that's it. And then you're going to click live update. What I like, what is different is that you can actually see the result in live with Elastic. And for example, if you see that here it's a background, you're going to just click a tiny bit. It's going to update the map directly. You can also select what you see and what you don't see. For example, in my case, I just want to see the cells, and you can actually increase the intensity of the cells. So you see that the map shows at the back. You see some blue in the between here. Here, I just want to tell him, oh, this is clearly background. So I'm just gonna do that, and it's gonna clean up the map. You can add another label. To do that, you need to stop, and I'm gonna just put foci. And then you can go back to update. I'm just gonna check. So for the foci, I know that here it's a foci. And but you see that it thinks that there is other foci somewhere else. Well, the probability is very low, but it's still very some. So you can actually correct them by telling it, oh, this is not a foci, it's actually a cell. And this is gonna better refine what is a cell, what is a foci. At the end of the day, what you have is a segmentation map, the same way that we did before. Okay, so these are a different tool. Then you can export the prediction as a binary mask, which is what we want to do. And you want to export like that. And it exports this directly into the folder that I selected here and save the files as well here. Oh, it should be able to export. Maybe because I am still in live mode. Oh, yeah, you put the uncertainty, the probability here in the folder that is a parent folder. And you can also batch process. So now that you have a model, you can select all the files you want and process them exactly the same way. So for example, this one, and this one. And then you can see process all file. And it's gonna segment them using the same algorithm that was used for the training. So that was Elastic, which I think is a great tool. It's gonna be refined. The last maybe what I want to show you is called QPath. It's similar, but it's made for pathologists initially. It does the same thing in a different way. You can bring your image directly by putting them here. And you can create a project so you save data inside. For the pixel classification, actually pathologists usually they have predefined classes. So you can just select this one tool, which is a wonderful tool. And you can select different areas, such as this one. And you're gonna say, okay, this is actually my background. So I'm gonna say set class background. I think one of the great tools in the QPath is the wand. The wand really knows what to select, and it's gonna. I don't know how they do that, but it's a great tool. I don't know how it works, but this one makes a big difference. So now I have my two things, and I can go and do my pixel classifier, classify pixel classification, train. And you can do live prediction. And this is what it looks like. And you can actually add and it will refine the prediction based on what you selected. And you can also decrease the display of this thing. And you can maybe select this guy and I'm just gonna say this is a set class. It's a tumor. And it's gonna train directly in live what you have been selecting. So I think it's a good tool. The same thing, you can export them The only downside of these two softwares is that they are not directly meant for extraction. So you have to kind of build your own script if you want to do extraction at the end. Okay. Any questions on that? So these are also based on machine learning and other resources for segmentation. I give you an example of Weka, Elastic, QPath. There is also other things such as Napari, Cell Profiler, which is not directly machine learning and pixel classification, but you can combine them with this. And then there is this brand new thing that has been released in April, CellPose. I didn't give you the demo because it's a bit more complex to install, and you cannot do the demo yourself at home. But it's super tool for pixel classification. Also, it works on human input to refine the classification, because you cannot. Initially, the project was making one single model for everybody using a huge amount of data. But then they say, oh, we can actually refine it by kind of narrowing down according to your specific data. So that's what they did, CellPose 2, that has been released recently. I tried it, it works very well. And there is a web version for CellPose 1 where you can actually just drop your files and they will do the classification for cells, for nuclei and this kind of thing automatically. So you don't have to think about it. And so that's a great tool. The last topic of this session is about extraction. And the good news is that it's actually fairly easy. Once you have done the cleaning and the segmentation, then the extraction is only bring back the segmentation region of interest back to your image to extract some data. And there is a lot of data you can actually obtain from pixel values. And these are very typical data, like the maximum within a specific region, the minimum, the average, the mean, the variance, the surface, the shape also of the object you discriminate. And all of that are very important, and they are kind of summarized in this web page for all what is image related data. So all the measurement, they are very well defined in this web page, and this is what you extract. How I usually do is I extract all of the data because it doesn't take too much space on your hard disk to extract everything. And then we have actually statistical model to analyze multiple variables at the same time. So when you don't know what to look at, and it's sometimes good to have all the data, and then you maybe gonna narrow down afterwards. Yeah, that's the conclusion slide telling you that what you need to know about data processing, the pre-processing, the segmentation, and the extraction. This is really give you the take-home messages and also the hands-on experiment using the demo I've done. I put other resources available, such as libraries for image data, where you can go and pick up things. There is also other tools, but I have to tell you, I didn't have the time to go in depth to present to you, but it seems that Jupyter Notebook is a big hit. KNIME also is a big hit, but it's going to be part of our research life in the near future. With this, I thank you all, and we have maybe a short break before we go back to the last session, which is automatization. And I want you to play with your own data and tell me, okay, I want to do that. How do I do that? And yeah, if you have your own data, then it's the time to open it up and bring it up. And you have a 5-minute break before we go to the next part of the session. Est-ce que je peux te mettre en pause de recording? Je ne sais pas comment on fait ça. C'est bon? Ça a redémarré. Alors, on va recommencer la l'atelier. Et la troisième séance, c'est sur l'automatisation. Je vais faire juste un très rapide overview de ce que c'est l'automatisation, mais après, j'aimerais qu'on aille jouer avec des données. Donc, je vais partager mon écran. Est-ce que vous voyez mon écran? Oui. Parfait. Donc, on a fait les deux premiers. Merci. On a fait les deux premiers points qui sont les images numériques, le traitement d'images, et le troisième, c'est l'automatisation. Alors, pourquoi automatiser? Pourquoi et comment surtout? La plupart des gens me disent « Ah ben, automatiser, c'est parce que c'est beaucoup plus rapide que de le faire à la main. » Yes and no. Doing automation for analysis is not faster than doing manually because it depends. It depends of how many images do you need to process. It also depends how many times do you have to do this analysis. And also it depends of your skills, how much time it will take for you to code this. And because most of us don't have a lot of programming skills, usually it takes a lot of time for people and they rather do it manually. But this is not the case for all of us. And most of the people that are doing programming, they're also into sharing a lot. So they write codes and then they make it available, which is a good thing for people that are not programming because they can take part of this code and maybe read a bit about them and modify it. For me, automation is very important because it's more reliable and reproducible than doing things manually. Also, it's trackable. If you do an automatic processing of images, you can know exactly what you have done. And if someone else wants to redo the same thing, it can do. You can just send them the process you have used. Usually, it's not biased in the sense that the machine doesn't have any personal interest into changing the data. So it just shows you the data it's making, and that's it. One other thing that ImageJ and Fiji can do is record macros. And for most of us, this will be sufficient for doing things that automatically. So I'm going to give you the example of how to record a macro, but this is not. This is just to give you the kind of the basic for programming. So I'm going to go to ImageJ here. There. I'm just going to close these windows. So if you have your ImageJ, here you have plugin. Oh, vous voyez plus mon écran? Oh, OK, ça va revenir. Share screen. Est-ce que là, vous voyez mon écran? Woohoo! Donc, si on va dans ImageJ, if you go to plugin and macros, and here you can have a record. And this is a macro recorder, and this is how I learned to write macros in ImageJ. No kidding. So anything you do in ImageJ is going to be written in the macro tool, what you have done. So if you select object, it's going to write this, what you have done. If you open an image, I'm going to open, for example, blobs again. You see that you run blobs, and this is actually the command line that sends the information to ImageJ to do this task. If you threshold the image, for example, adjust threshold, you see that you can actually modify your threshold into something that is relevant to you and click apply. And then you see that in the recorder, it shows what you have done. And then you can close the image. And then you can click create. When you click create, it shows you the macro that is has been prepared for you. And one of these, just one thing about macro is everything you put with 2 slashes is comment. So it's not being made. So you can actually erase them. It won't change anything. So this macro, the first thing, it opens blobs. It does an auto thresholding. It runs the threshold, but you see that this has been in a comment. You can actually set the threshold automatically, and you can convert to mask, close the file, and that's it. So if I run that now, I remove the close, I should open blobs and do the thresholding itself. But most of the time, you don't want to do things with blobs, you want to do it on your own file. So you can actually open a file such as the DAPI that I was using before. So you open a file like that, and then you can say, okay, now I want to do the same process here. So I'm going to remove random blobs, and I'm going to threshold, and that's my result. So if you have tasks that are kind of very time consuming, you can actually script them and do that. You can also write a small script for example, we're going to do a filter. And then I'm going to tell you how to apply this filter to a full folder. So I'm going to open blobs. And let's say I want to put a Gaussian filter. And my Gaussian filter is going to be 22. So it's all blurry. I want to apply this to a bunch of files. So I'm going to create a macro. And I'm going to just use run Gaussian. I'm going to save this macro into a Gaussian.igm on my desktop. Okay. And now let's say I want to apply all of that to a folder containing pictures. And here I have a folder containing pictures. It's on my desktop. So I can go to ImageJ, process, and batch macro. I'm going to say input. Input is in my desktop, and it's a DAPI for QPath, open. Output, I'm going to create a new folder called output on my desktop, open. And then I'm going to say, actually, I want to put this macro here, Gaussian. So I'm going to say open. I'm going to select the file that is called Gaussian, open, and process. And there is an issue. I don't know why. Maybe he tried to open these files and he didn't. He was not able to open them, so that's why there is an error. But these 4 files should be processed. So that's a very quick and very easy way to automatize things. I have been asked by some people to do some kind of data analysis. Where is that? No, that's there. So if I go here, I think Florence was contacting me to do something quite simple. So she has these files here. And she told me these are, I'm going to just tile them. So I have my picture. This is one picture, but it has 3 colors. I want to merge them. If you have only one picture, the easiest way to do it is to actually do color combine and merge channels. Oui, c'est vrai. Donc, well, because they are RGB images itself, you can actually convert them to maybe 16-bit images first, and then merge them. And then merge them using color merge, and you can choose whatever color you want. Done. You can do that, but you can also write a script to do that if you have to do this often. And it just happened that I wrote a script for that a few years ago, and I put it on my GitHub. So I wanted to you guys know that you can actually search for things and get them from other people. And this is very trendy into the ImageJ world. In Fiji, you have a lot of plugins that have been written, but people like me also makes plugins. And I'm not very good at writing, but sometimes they are very useful. So if you go to the GitHub, you're gonna go there. You're gonna search for, oops, I don't want to sign up. I want to search here. You're gonna search for Stephanie. I think my name is Anne Stephanie, but doesn't matter. Yeah, I'm here. And then I made these things called spinal cord mapper, which has a function to do that. And to do that, you can just download this jar file. And a jar file is just a zip file, but with an extension that is called jar. And if you put it into ImageJ, it's going to install it there. And then in your plugin, you're going to have the plugin showing up. And I have this thing that is made for merging images. So I'm just going to show you what it does. So you click OK, and then you select the folder containing the files you want to merge. And I put them on my desktop into the input folder. These are the files that has been sent by Florence. He asks you how many channels you want to merge. There is 3 channels. And then it is say, how do I identify my channels here? Well, my first channel is a DAPI, and it's finishing by D0. My second channel is finishing by D1, and I don't know, it's a neuron. My third channel is D2, and I guess it's Nissl staining. And then you press OK, and it did the processing, and it saved the file into a new folder called input merge. And then you have your image. So for one image is meaningless, but if you have a bunch of images, or if you have to do it many, many times, then maybe it's worth doing it. So it's going to save you time over. It's going to save you time over time. The second thing she asked me to do is to remove noise, and I guess we saw that it's possible to remove noise quite easily. There is quite some noise into the green channel and in the blue channel. So one of the ways to remove noise is to do process subtract background, and I have a dark background this time. So the features I want to look at are quite small. So I'm just going to make sure to use a sliding parabole and 50 pixel diameter. And if I do that, then I clean up a lot of the data that is out of focus, and my data looks much better. So you could apply that in batch to all your files, and that should be fine. So that was a specific example. Just to let you know, if you want to learn about coding, you can steal what people have done before. And in this case, if you want to know what's the code that will allow me to do the merging, for example, the code is quite long because I made it for being useful for many, many people. But you can actually just click on that, and then you have the full code here. It's a very simple function, but if you want to make it work with all of possible cases, then you have to be a bit long. So you can actually just copy that or copy part of it. I usually write precisely what each function is doing. So you can actually take this part of the code or some of the code, or copy and paste some section. I don't want to go into the details because it's a very complex code for doing something very simple. But in this case, it's a bit, he has to look for patterns into the names of the file to find the file that have the same name with a specific suffix, and then kind of merge everything together. And I wanted to make it work for a lot of people. So that was one example from Florence that was telling me she wants to do that. Maybe one exercise was coming from someone called Lydis. Lydis told me she has some files and she wants to segment them. So where is Lydis' data? And she sent me quite a lot of, images. And each image is it's actually a big file. It's 38 megabytes because it's a stack. So maybe one exercise for you guys would be to actually write a code to do a maximum intensity projection of all of these images in batch. That could be an exercise if you want to try how to do that. So we don't need to have a Z stack for all of the images. You can do a Z projection, and to do the Z projection, you just go to stack, Z project, maximum intensity projection, and BIM. You have your data that is now in 2D. Much better. and it's 1 megabyte only, so it's going to be easier to do the segmentation. In her case, she wants to identify cells that have foci versus non-foci, so we could segment them and then do the extraction of data. All right, so now we're going to go to your data. Anyone wants to share a screen and show some data, or tell me what you guys want to do, and I will tell you how to do it in the best way, in my view. Or if you wrote a plugin yourself, or if you know someone who wrote a plugin and you want to make a demonstration of it, you're also welcome. I didn't mention it, but there is a microbe G that was written by someone at the Université de Montréal that is specifically designed to look at the shape of bacteria. So that's a full plugin also that has been that is available. So anyone interested in taking over, or sharing, or looking at data? All right. Then what I can do is show you another thing about the spinal cord mapper. It's actually a very versatile tool. So I installed the jar file, but this also have a toolbar called spinal cord toolbar. So if you write a bit of code, then you can actually make a toolbar to have shortcuts directly to your image. And also for spinal cord mapper, I made a demo data, so you can actually download the demo data directly on GitHub. So a lot of people have a GitHub website like that. So you can just download this. I am just one of the many people, and not the best guy to program, but just to show you a demo of what you can do. You can merge images, and this you saw what it was able to do. Desktop. In this one, you see that I have 3 channels per slide, and it's a suffix C1, C2, and C3. Okay. So it processes it, and now I have my 2 images. One is this way, and the other one is this one. Oh, you see that my 2 images are not next to each other. Maybe it would be cool to align them. And this is a process called registration, or orientation, or alignment. I made it in 2 ways. There is a simple registration. So if you click on this, you can actually select a full folder. And I'm going to select the one that are merged. And the simple registration is you just draw points into your sample. Actually, it's this guy. At defined places, but in this case, I define them myself and say, okay, I'm going to make 7 points around the spinal cord. And these points are always the same, no matter which image I'm using. And it opens the next one, I do the same thing. And there is one too many. Oh, yeah, I thought. Oops. Forgot one. And when you finish the process, you should have an image that are registered. And they should look like that. They just are the same image with the point on them. You can actually align them also. Oops. Same thing, but this time it will align them one to the other. this is useful when you have several images and you want them to look all the same at the end. Damn it, I forgot always the same one. And if you look at the result this time, it should be fairly aligned. Done. So you could actually put them into a stack and look at them, and they're all well aligned. Then there is usually something that people want to do is count cells. So I'm going to do this on just one example. There is two ways of counting cells. There is the manual way. It doesn't matter if it's manual or automatic. What matters is that it's traceable. You can actually remember what you have been counting. So in this one, it just show you the counter, show you each channels individually, and then you can count cells, 1, 2, 3, 4. And then you go to the next counter, you say, oh, there's 1, 2, 3, 4 here. And maybe on another slide, I'm gonna put a new counter here. I'm gonna say, oh, this is my over neurons. But what this does is just it keeps track of things that you have done or you have counted. And you can give them a name to the counter. And at the end, you have the region of interest, you have the data, and the result of each point you draw. Another thing so you can use is auto cell detection. And this basically do the thresholding and everything, but it doesn't include pixel assignment because this was done 5 years ago. Actually, I didn't look at it since 5 years. I wrote it into a lab when I was a postdoc, and then I forgot about it. And then I said, oh, maybe it might be useful, at least for some part to someone. Yeah. That was an example of what we can do, but now it will be nicer if we have data from someone that want to share a screen or anyone interested into doing real data, real things, real science. So many. On peut les regarder ensemble sur mon ordinateur et partager mon écran si tu veux. Tu peux prendre une chaise et si vous voulez, on va regarder les données de Isley. Excuse-moi. PC en Mac. Hop, je vais mettre les 3 images.
Ce sont les mêmes. Je crois que la dernière.
OK. Je vais partager mon écran et on va regarder juste le travail qu'on va faire avec Hailey. Tu veux prendre une chaise? Hailey a 3 images qui sont déjà prises en format qui s'appelle le format RGB.
Ouais.
So RGB format is not very good when you have to export. It's okay that colors are there, but RGB are not really good. But the good thing is that in this case, you only have R and G.
Yeah.
So if I transform this image into a TIFF, a multi-layer TIFF with several channels, this should be fine. And how do I do that? Well, for this thing, you need to first color, and you need to split the channels. And obviously, you have the blue channel is empty. And then you have a green channel and a red channel. And then you want to recombine them. So you're going to say color merge. And you're going to say my red is red and my green is green. And create a composite. So now I have a 2-channel image. I can look at the channel tool. I can look at them in a grayscale. And I have these cells, and some of them are stained. So I'm going to save this image into my input. And this is the number 1, but it's a composite. And what do you want to do next?
Localization.
So you want to look how much green is in the red or how much red is in the green?
Green, red.
So you want to know how much?
Normally it's to identify the red, where green it's in the red, because all of this is crab.
Yeah, crab.
So you can localization.
So can we crop into a specific region of interest?
Yeah.
Oops. I clicked too fast. Oops, I clicked too fast again. What happened? So we're gonna just crop on this image, and we want to know how much green is in the red.
Yeah.
So this is easy. I can draw a region of interest here within the green.
Mm-hmm.
And then I'm gonna add this to my manager. So I go to RI manager. And I'm gonna click add. And then I'm gonna do another region right there where it's red, but I see it's not green. So it's just an area just next to it. And I'm gonna add. And then I want to measure what is the intensity within this region versus what is the intensity within this region. And that's what we do. So you see the mean intensity in the first region is 70.
Yeah.
The mean in the second region is 92. So you have less red in the first region and you have more red in the second region. Then we're gonna do the same thing, but in the second channel. So I move to the second channel, measure 1, measure 2. And here you see that you have much more green in the first channel than in the first area than in the second area. And I guess you want to do this in an automatic manner because you have many. Yeah. So you will have to segment the image because it's not possible to kind of draw a single region of interest.
Yeah. For me, I created the mask to create CTB.
Yeah.
And then after I create another mask with the 2 green, and I merged them.
Mm-hmm.
So it gave me only mask is in the red.
Okay, but
so you can determine the threshold, not threshold, but the density of green.
Okay,
but I wrote the macro.
You have it?
Yeah.
You want to show?
I don't remember where is this, but
okay.
I can show you another time.
Okay, but the concept is that you take the green,
yeah,
and you threshold it.
Yeah.
And with this, you create a mask. So
mask. Yeah.
I don't know what is positive and what is negative, but I guess something like that is positive. Or we can use maybe.
I can determine the threshold. I determined the threshold, and then I create the mask.
Okay.
To fall.
And then we're gonna create a selection. Edit. Make selection. And then I'm gonna come here and I'm gonna say selection.
I created
restore selection.
1 year ago, I don't remember.
So I took my mask and I create a region of interest of everything that was green above a certain threshold. And then I go to my mask where I have all this green, and effectively I can see that I have green. And what I want to measure is how much red is inside.
Yeah.
Right? So I'm gonna just do the measurement. And then I want to know how much red is in the other side, like in the place where you don't have green. But in this case, you have to be careful because there is 2 places. There is the green that is the red that is within the cell, and there is the red that is outside the cell.
Yeah.
So you need to also create a mask for the red one to select everything that is above, that is actually a cell, and we have to measure everything that's within this cell. So I'm going to duplicate the first channel. like that. And I'm gonna do another thresholding.
Yeah.
And again, something like that seems to be reasonable to me. More like that. I don't know what we are looking at and if it's a molecule or if it's a full cell. And then I'm gonna do the same thing. Edit selection, create selection, and here, selection, restore selection. And now what I want to measure is how much red is in my red. And then I want to compare these two numbers. So in my first level, I have a big area and my level of red was 47. So I have 47 on average intensity of red within the green. If I go outside of the cell where it's mostly red, then I have 100. So it's redder outside of the green than inside of the green. If you want to know how much colocalize, I guess there is a plugin for that. I don't do colocalization and I was planning to do it in the next session. So I don't know much about it, but
I have one question about the color threshold. You can make it one kind of colocalization, I think so. I remember.
Color thresholding? Yeah, I never tried that. Let's try.
See, it's called image color.
Maybe adjust color threshold.
Just color threshold, and then you can make you can determine the threshold of each color.
Okay, but for this you have to be in RGB because now we RGB, went back to the. Okay, let's try that. Never tried. Anyone has experience with that? So this is based on the brightness, the saturation, and the contrast.
It's here.
Yeah, so actually brightness is here.
Right.
Okay.
So you can determine or green or red.
I see.
Yeah.
I would say the other method is more reliable because you divide each channel and then you recombine them. So you analyze them independently of each other, which in my view would be better. Any question in the chat? Merci. In this case, don't we have subtract the green and red backgrounds before evaluating the density selective labeling? In the case of Islay, you want to subtract the green and the red background? Yes, we can. Before evaluating the density. You mean that this tissue around here contribute to the fluorescence you have here? Yes, this is a very good point, and this is true. You should, or you may, but the question at the end is, what does Islay wants to look at? If it's a intensity that is normalized for his tissue, I would say yes. If he's looking at a relative intensity between the two, maybe not. If it's a proportion of pixels that are normalized, maybe not as well. But this is a very good point. You could take the value, the average value that is outside of your region of interest and decrease it by this amount. So you will remove some noise that is outside. A good point. J'en ai pas parlé du tout, mais si tu veux en parler. According to Monique, and I think it's a good point, the best way would do to have a negative control of a slide that is not stained with a primary antibody, but only with the secondary antibody. Measure the fluorescence that is located there, which is really related to the background, and then subtract this value to the image.
I did it, but it's very negative.
Well, that's very possible. Another thing I looked at was, I told you, you know, if some people knows a bit about scripting, they could write a script for making the blobs. Is it blob? No, it's not blob transform. I made a little macro, and I don't know where is it. the piece, cell pose test, blob transform. I made a little macro to show you guys, but oh, here. In the middle of a talk, I said, oh, if you can write a small macro to do something automatized. And this is the macro I wrote last night. It's interesting because you can use the record function to kind of know each of the function that is here, and then say, okay, you're gonna do blobs, and then you run this function, and so on and so on. But it's actually shorter to kind of make a for loop. And I say, okay, I'm gonna make a for loop for each iteration of this. I'm gonna run a new function that is part of a list of functions, if you want. So if you run this, maybe because it's new format. Yeah, it was written for image J1, and I opened it with Fiji. So if you run it, it opens the blobs. It does the function for each of them, and then also it used this. Actually, I have your image here. It also, oops. It also put a montage, which is a magic montage to show you all the images together. So that's all the features you can use with ImageJ, and the name is at the bottom. So I will put the script, the macro into the course, so you can take part of this. And if you want to have help to understand how it works, I will explain to you. But in the bottom line, these examples, there is many of them online. You are not alone to ask yourself these kind of questions. So most of the things that already has been done. It's just a matter of finding where it is. If you don't find it, or if you need help to find it, you can contact us. And that's it for today. I think it's time for food. Thank you all for coming. And I want to tell to the people that are here that there is a barbecue made by the biochemistry department. It's going to be on the Louis Collins parking lot. And it's going to be very, very cheap burgers at cost, so you can come. Thank you all for your time today. Any questions, please send me an email, microscopyfacmed@humanmaterial.ca, and I'll be happy to answer and to also to get your feedback.
Bye.
Bye.
Como YouTuber.